Hello everyone, my name is Renan Lima and I would like to share with you a little bit of my PhD work, which was conducted in the context of the COMNAP Antarctic Fellowship. The title of the research is Ecological Divergence in a Climate Change Sentinel from the Antarctic Peninsula, Evidence from Bulk Tissue and Compound-Specific Stable Isotope Analysis. So the Antarctic Peninsula has been under complex temperature regimes which vary temporally and spatially. The Western Antarctic Peninsula is characterized by reduced sea ice extent and duration in the long term, while the eastern side has permanent ice and only recently significant changes were detected. Warming in the northern and western Antarctic Peninsula has affected multiple levels of the food web, and examining the differences in the foraging ecology of sentinel marine fauna between the two sites could serve as indicator of how other populations can be expected to respond and inform conservation management. So, measurements of relative abundance of naturally occurring stable isotopes are useful tools for characterizing food web dynamics and structure, whole tissue, which are referred to as bulk tissue from now on, stable isotopes of carbon, which are reported as delta 13C, and nitrogen, delta 13N, reflect the diet, habitat, and trophic position of individuals. Carbon is transferred up the food web with little fractionation, while nitrogen becomes more enriched in the heavier isotope at each trophic transfer. Carbon is then usually used to discriminate between different primary productivity sources, and nitrogen as an indicator of trophic position. Although it can be also baseline influenced, carbon differences are also expected between pelagic and benthic producers. So, isotopic analysis of individual compounds such as amino acids provide an enhanced way to disentangle baseline from trophic effects. Amino acids can be classified as either essential or non-essential. The carbon of essential amino acids cannot be synthesized by consumers and are passed along the food web from primary producers without alteration, while non-essential amino acids can be synthesized de novo. Nitrogen isotope dynamics of amino acids follow a different categorization, with amino acids classified as trophic or source depending on how readily the nitrogen can be trans or deaminated. The delta 15N of source amino acids normally undergo little modification, and similarly to the delta 13C of essential amino acids, reflect the isotope values of the primary producers at the base of the food web. Offsets between source and trophic amino acids allow us to calculate more accurate estimates of trophic position. So the Weddell seal is distributed between the fast and pack ice limits. This species is an apex and generalist predator, consuming pelagic and benthic fish, cephalopods, and occasionally crustaceans and other invertebrates. Sexual size dimorphism is low, but different foraging habits were reported for some populations. So, assessing trophic dynamics is important to understand the resilience of ecosystems under a changing environment, since changes may affect the energy transfer and negatively affect species survival. Here we analyzed bulk tissue and compound-specific stable isotope analysis in whiskers of a climate sentinel from two sites differently affected by warming. Our main goals were to evaluate temporal, sexual, ontogenetic, and spatial differences in Weddell seal's isotopic values, and determine if and how baseline and diet differ between Eastern Antarctic Peninsula and Western Antarctic Peninsula. So sampling occurred in close geographical proximity but under different ice and oceanographic conditions. In the west, sampling occurred near Primavera Base on the Danko coast, while in the east, sampling occurred in the proximity of Marambio Base at Seymour Island. All logistics and sampling were carried out by the Argentine Antarctic Institute. So, hauled out seals were sedated. Whenever possible, animals were sexed and measured. The longest whisker was plucked from the masticial pad of each individual and stored dry. Whiskers were cleaned and sectioned in 5 mm segments, of which a 0.2 to 0.5 milligram randomly selected subsegment was weighed in thin cups for bulk stable isotope analysis, and the material left was stored for compound specifics. The number of whiskers subsamples for amino acid analysis ranged from 1 to 4, and 21 out of the 52 whiskers were used for amino acid analysis, 9 from the eastern side and 12 from the western side of the peninsula. The size of the sections varied according to the mass required, from 2 to 10 milligrams in the whisker size. And after hydrolysis and derivatization procedures, which break the peptide bonds and make amino acids volatile, amino acid samples were analyzed on a gas chromatograph coupled to an isotope ratio mass spectrometer at the University of New Mexico. Due to logistical constraints, only part of the samples analyzed for amino acid nitrogen was analyzed for carbon. We used linear mixed effects models to assess the relationship between bulk tissue carbon and nitrogen separately, with site, sex, year, and total body length. Additionally, we employed the cyber package to generate isotopic niche metrics as a proxy for the ecological niche, and to determine niche overlap between groups. We also conducted the principal component analysis to detect spatial patterns of amino acid isotopic composition, and finally we utilized an equation modified from Chikarashi et al. Based on proline and phenylalanine to calculate trophic position. So bulk tissue delta 13C was higher for the eastern compared to the western Antarctic Peninsula. It is important to highlight that carbon values from the eastern side were very anomalous compared to other values from other pinnipedes in the Antarctic Peninsula, including the Weddell seal itself. The western individuals, on the other hand, showed similar isotopic values to other populations, such as the Ross Sea and the southern Weddell Sea. Based on the 40% ellipses, we found no isotopic niche overlap between the two populations. In the western Antarctic Peninsula, males showed higher nitrogen values than females, which corroborates previous results that suggested that males explored 15 nitrogen enriched shelf areas, whereas females are more pelagic. We also detected a significant decrease of delta 13C in this population, considering both sexes. In the northern and western Antarctic Peninsula, the replacement of diatoms by cryptophytes due to ice melting and water column stratification could contribute to the observed baseline differences between Weddell seals, since the smaller cryptophytes are 13 carbon depleted and diatoms are 13 carbon enriched. Furthermore, foraging sites' baseline values of Weddell seals could also be under influence of different CO2 dynamics. The increased upper ocean stability due to the recent warming has led to higher CO2 uptake by phytoplankton and higher biomass in the western Antarctic Peninsula. The higher uptake of CO2 by phytoplankton allows higher incorporation of the light isotopomer, preferably used during photosynthesis, leading to 13 carbon depleted delta 13C values. In the amino acids, we saw consistently significant differences in both essential and non-essential amino acids, except for tyrosine. When using amino acids, it was evident that they occupied the same trophic position, and nitrogen differences noted in bulk tissue values were mostly driven by trophic amino acids, suggesting that even though populations have similar trophic positions, they could still have different proportions of prey contribution to their diets. When considering the amino acids, we also observed strong segregation between the populations based on carbon, likely due to different sea ice conditions between the regions and its baseline influence. So it is possible that the eastern peninsula seals are foraging in areas where 13 carbon enriched sea ice associated phytoplankton predominates. The extensive ice cover might also affect the dissolution of atmospheric CO2 into the water. So if less CO2 is available for ice algal growth, they would take more 13 carbon, which would explain the 13 carbon enriched isotope values. Identifying the source of the 13 carbon enriched isotope values could provide further insights into the foraging habits of the Weddell seals in the eastern Antarctic Peninsula. However, this is challenging considering our limited knowledge about this area. Regarding nitrogen, small differences related to the diet seem to exist, but they are not as significant as the differences observed in carbon. So some take-home messages are: there's significant foraging divergence between Weddell seals from the eastern and western Antarctic Peninsula, which seems to be baseline influenced. The higher sea ice cover in Weddell Sea compared to the western Antarctic Peninsula likely has a strong effect on phytoplankton community structure in CO2 dynamics, which is propagated up the food web. And examining the differences in the foraging ecology of sentinel species between areas differently affected by climate change could serve as indicator of how other populations can be expected to respond, such as shifts in energy sources, and as well as ecosystem resilience. Despite the significant carbon differences between the eastern and western Antarctic Peninsula, the western seals appear to be less sensitive to climate change compared to other seals and exhibit similar isotopic compositions to other populations. In fact, they have demonstrated dietary plasticity in the past when colonizing ice-free areas. So further exploration is necessary to understand the ecosystem characteristics of the eastern peninsula that are responsible for the unusually enriched 13 carbon values. This exploration could be achieved through the use of animal-borne instruments or dedicated ship service, which would enable a deeper investigation of the biological and physical features in this area. So I would like to express my gratitude to COMNAP for granting me the 2021 Antarctic Fellowship. Thanks to this fellowship, I had the privilege of spending a few months in the beautiful state of New Mexico in the US. During this time, I had the opportunity to collaborate with Dr. Seth Nilsson and Dr. Nico Lupker on amino acid stable isotope analysis. And this research was part of my PhD dissertation in biological oceanography at the Federal University of Rio Grande in southern Brazil. And working with Dr. Nilsson and Dr. Lupker provided me a unique learning experience and valuable insights into the isotopic field. And it was particularly significant for my career growth, as there are currently no labs or universities conducting compound-specific analysis in my home country. And I believe that this fellowship is making a significant contribution to the training of polar scientists in new areas of expertise, which is particularly important for scientists from developing countries who are naturally exposed to fewer opportunities and have limited access to cutting-edge technology. I would like to thank every institution involved in this work, and here's my email if you have any questions or would like to get in touch. Obrigado.